Hi, good morning. This is Miss Dagenford, and we're going to be looking at translation, which is the next step of our protein synthesis. And if you recall, we had our central dogma, and the central dogma started off with DNA, and DNA is transcribed into mRNA, messenger RNA, and then it gets translated into a polypeptide. Now, we mentioned in class that there are other things that DNA is going to be transcribed into, but we're going to look at the polypeptides for now. So let's look at some of the players involved. With translation, one of the first things you need to understand is transfer RNA or tRNA. And so by the end of this video, you should have a good sense of all of the molecules that are involved in translation, the processes, how it happens, and then some of the ways that we can regulate, or at least start talking about regulation. So here's our tRNA molecule. Now, the first thing you need to understand is this diagram, which you often see in textbooks. This is the schematic. If you were to unravel the RNA involved in transfer RNA, you could lay it out in a way that shows that they bond together. So here's our hydrogen bonds between our bases, the nucleotides, where the little dots are. Those are the hydrogen bonds. And then you have these little loops that don't bind. There's no binding here, so they bubble out a little bit. Now, that's all fine and good, but that's not what it actually kind of twists into. Down here, this is more of the accurate model of how a tRNA looks like. So there are several regions on the tRNA molecule that are of interest to us. One is this end region, which is where it will connect to an amino acid. And this is the appropriate amino acid. So there are multiple kinds of tRNA molecules. Then the other end would be the anticodon. Now, the anticodon matches or complements the base pairs that are in a codon that's on an mRNA strand. So, for instance, and I know this might be hard to see, but if our anticodon is AAG, then the codon it matches must be UUC. And then that's how we're going to uh, relate the correct amino acid to the correct codon. So sometimes I like to think of this as not transfer RNA, but translates, right? It translates, and that's why we call it translation. So this is the actual shape it twists into. So here's our anticodon down at the bottom, and here's the connector for the amino acid. And then oftentimes in books, you'll see more of a symbolic version, a symbolic model, and this is what you get, where you have that little bent shape that occurs. And again, here's our anticodon, here's our codon for the mRNA strand. So how does this work? Well, the first thing we need to do is actually start the process, and that's called initiation. And so down at the bottom, you see the small subunit of the ribosome. So this picture, let's go over what this indicates. The brown is going to be the ribosome, and this is our large subunit. And you recall from our cell unit, the ribosome makes protein. So here we are getting into the specifics here. So we start with a messenger RNA strand, the small subunit, and notice it'll bond together with the tRNA molecule with its appropriate amino acid. So this little hexagon up here, that's an amino acid. Here's our tRNA. And then there's our small subunit. Once that small bundle, that small complex, can all connects together, we need an ATP molecule to start it, and then we have the large molecule coming in. The regions of the ribosome are important to understand. We've got an exit site, a P site, and an A site. Now, there's the common name and then the fancy name. The P site is named after the fact that you have the growing peptide chain, the growing polypeptide coming out of this P site. The fancier name is the peptidyl. And then the A site is when you get a brand new amino acid coming in, thus amino acid or A site, but we also know it as amino acyl. So there you have the EPA, all of those sites. So how do we then move on? So this is initiation, and then we have elongation. Now elongation, and this will definitely come together when we watch an animation of it. But you have the ribosome with the mRNA strand. Now I went ahead and labeled these codons. They, I'll be honest, they don't match up with the shapes, so don't worry about those for right now. So let's say we've got 1, 2, 3, 4 amino acids already, all connected together with a peptide bond. Well, let's say that this is CCC, and then you can imagine what I might ask you. If this is CCC, then what's our anticodon? And then let's say the next codon is UAC. Well, here we'd have our tRNA molecule coming up to this spot, and it must fit. Those complementary base pairs must fit together between the codon and anticodon. That's where we have the matching. So we have a new tRNA molecule that arrives at the A site carrying the appropriate amino acid. Now, this doesn't happen spontaneously. We need some energy, and so that's going to be our ATP molecule there. Now, the job of the ribosome is to catalyze that new peptide bond between the new amino acid and the existing polypeptide. So you see that happening right here. Here's that little bond occurring, that peptide bond. Once that occurs, then you can get rid of this bond that holds the amino acid to the tRNA molecule. So you'll see what happens next. Now, keep in mind, I've labeled everything for you. So notice as it shifts to the bottom picture, here's the bond breaking, and there is going to be a shift that occurs. Again, we need another ATP molecule that helps this happen. So as we switch from the bottom picture to the picture on the left, notice what happened to the CCC here and the UAC. They are now moved to the left. So this is called translocation. The ribosome actually moves down the mRNA molecule. And so the tRNA molecule that does not have the amino acid, it exits to the exit site, and now we have an empty space in the new A site. Now that it's empty, you can guess what happens. You'd have a new tRNA molecule bringing the appropriate amino acid, catalyzing the bonds, breaking the bond to the polypeptide, and then the tRNA molecule leaving. So in this way, what you have, as long as it keeps going, you're going to continue to grow that polypeptide. Now, eventually you get to the point where you have a stop codon. And for a stop codon, there is no appropriate tRNA molecule. You just simply have a termination factor. And the termination factor just causes that ribosome to just spin its wheels, and that loosens all of the bonds. And then you have an ATP molecule coming and allowing it to break off, and it can go ahead and translate another mRNA molecule. Now, what you want to be thinking about is this is where we connect it back to the DNA. This is where we connect it back to the cells and the different cell types. Suppose you have a pancreas cell. The pancreas cell is going to be making lots of insulin, a small polypeptide. And then a B cell in our immune system is going to be making a lot of antibodies. Now, each one of those proteins comes from a different gene, which means it comes from a different mRNA molecule. So, how does each cell, quote unquote, know or get that signal to make that particular gene? Why doesn't that pancreas cell make the antibodies? Or why doesn't that B cell make the insulin? So it gets into this idea of transcriptional controls or translational controls. How do we tell that cell? How do we signal for the different genes to be made? So, one thing I want you to be thinking about is anything at all that allows for RNA polymerase, which is that earlier transcriptional stage. anything at all that makes that happen more easily, or it's more likely to happen, that's going to increase the likelihood of that mRNA being transcribed. So that's going to be our next step in looking at how our gene regulation is occurring. Well, that's going to be the first step. The next step is going to be talking about some of that. So you've got a short video here, and then we're going to have a longer video next. Have a nice day.